Hello there guys, Teacher Krishna here, and welcome to our next class for the third unit. Of course, this is a class that is part of BHEV School and 9th BYS. Our subject, just like you know, is biology, and today we are going to be working on the content for week number 24. So let's go over our presentation for today. It's very important to remember to have your notebook with you in order to make today's summary. And let me refresh your mind about the parts that your summary must include. Date, ending mind, title, the summary itself, and of course, some extra decoration. Right, and today we're going to be studying lesson number 3, and the topic is Advances in Genetics. You are going to find this topic in pages 146 to 151. Our ending mind for today is the following. Describe 3 ways of producing organisms with desired traits in order to create a mind map about hybrids. Alright, very important. Remember to keep track of your ending mind in the essay. So after you copy it, you can continue with the video. Let me give you a small summary of the activities that you need to complete. So watch this video and make a summary. Then read lesson 3 and complement your summary. I will suggest that you focus specifically on pages 150 and 151. But remember, it's necessary that you read all the lesson. Then complete the book's exercises in pages 6 to 151. And we are going to complete a couple of exercises just like every week during the mid-session. And we're going to complete the exercises in pages 150 and 151. So make sure to complete the exercises that are before those pages. Right. Lastly, take pictures and make a document with a compilation of your work. So take pictures of your summary, take pictures of the exercises in your book, and then share it in Google Drive. All right. So today you are going to make a folder again, but this time in week number 24, and there you have to upload your document, right? So let's go over our topic, and the title is Advances in Genetics. So what happens if you mate a zebra in a horse or a zebra in a donkey? Then you will get horses and sea donkeys. Well, probably sounds weird or something like science fiction, but in reality, I can tell you that this happens in real life. It's not that common to see, but it does happen. And this only happens when humans do it on purpose. But why will humans mate zebras, donkeys, and horses? Well, people try to get these new species to be more resistant to certain environments in order to commercialize them in different places around the world. So, of course, it's way more difficult for donkeys and horses to survive in African environments, especially. So what people are trying to do is that they are mating. For example, in this case, this is like a real picture of what happens when you mate a donkey and a zebra. So what people are expecting from this is that these new species will actually be able to survive in the African environment. So of course they can commercialize it and get some more money for their living. And I'm as surprised as you because I have never seen an animal like this one, but apparently they exist. All right, let's continue. So how can organisms be produced with desired traits? Remember, desired traits are desired characteristics that you would like to have in a certain organism. So here is a very clear example, and this is something that humans have been doing for a very long while. So what happens is that humans take a look at 2 species that they like. For example, in this case, we have 2 very beautiful dogs. So they say, oh, I really like the characteristics, or I really like the way these dogs look. So, of course, if they both have babies, then the puppies are going to be very similar to their parents. And that's correct. That's exactly what happens. When these 2 dogs have puppies, then the babies are going to be very similar to them, and which means that humans are going to get the desired traits in the offspring. All right. Basically, this is something that has been happening for a very long while, and scientists actually theorize that this is exactly the way wolves changed into other dogs. For example, how is it possible that from a wolf a Chihuahua was created, for example? Well, it was because of desired traits. So humans started selecting the ones they consider will look the best or will look better, and they will actually mate them against each other until we get the different races of dogs that we have nowadays. So there are three ways to produce organisms with desired traits. We have selective breeding, cloning, and genetic engineering. So selective breeding, it chooses organisms with desired traits to be the parent of the next generation. Humans use this with several animals and plants. So selective breeding is the example that I was telling you about. Choose two specimens that you like and mate them to be the parents of the next generation. An example with plants. Corn is one of the biggest examples. So many years ago, actually the corn as we know it didn't exist. It was like an ancestor of corn. But the thing is that humans, one more time, started selecting the specimens that they liked the most until they reached this specific type of corn that we have nowadays. And of course, since it was chosen and it was reproduced many times, it is the most common specimen of corn that we have right now. In selective breeding, we have two subdivisions. The first one is inbreeding. It means crossing two individuals with similar desirable characteristics, two dogs that are friendly. So I can tell you about golden retriever. So golden retrievers are usually very friendly dogs. So if you have a father and a mother that are very friendly, of course talking about golden retriever, when you mate them, it's most likely that their puppies are going to be very friendly as well. And also we have hybridization. And it means crossing two individuals with different alleles. And the resulting organism will have the best trait from both organisms. So actually, in this case, it will be different. It wouldn't be like two similar dogs, but they will be two different dogs. And of course, the most dominant allele is going to be the one that is going to be present in the following offspring. All right, we have cloning and genetic engineering. So this is an extremely interesting topic. And cloning is very helpful in order to create a medicine such as insulin that helps humans to develop a certain medicine to fight some disease. So let's go first of all with cloning. A clone is an organism that is identical to its genetic donor. And unicellular organisms are easier to clone than pluricellular organisms. So actually right now scientists have been able to clone some species. So the most enigmatic one was a sheep that was cloned. So basically the process of cloning is the following. So what we want is to create a copy of a certain animal, okay? So we need to take into consideration that we are going to be having two cells. One that is a cell that is already from a grown-up animal, and the other one that is going to be for the baby. So what happens is that we're going to have an unfertilized cell. And in this unfertilized cell, which of course will be how do you call this one? An egg. It will be an egg. It doesn't have enough genetical material to start reproducing itself. So what scientists are going to do is that they are going to extract the nucleus from that unfertilized egg. And inside of it, they are going to insert the nucleus of the cell that was originally from the other animal. So you know that the cell from the grown-up animal already has genetic information. And when they introduce it inside the other cell and it starts multiplying, they are going to create an identical copy of the animal. So that's pretty much what cloning is like. Okay. We have genetic engineering, and this technique consists in transferring DNA from one organism to another. So science is so great that right now scientists can actually separate one double helix of DNA. They can separate it, and they can actually insert another codification of DNA inside. So when the original DNA and the DNA from the donor combine, they are going to produce a different result. And after you read the pages in your book, you are going to notice that scientists actually created a glowing cat, which is something astonishing. And let's continue right here. It says scientists insert DNA from human to bacteria in order to produce large amounts of insulin. This can be used to treat people with diabetes who usually present a lack of this component. Just like I was mentioning, so certain substances can be created to support humans whenever they need to fight a certain disease, such as diabetes in this case. And in our book, we found this process specifically on how people insert DNA and how insulin is created. So let's go over each one of these steps. So number one, we have small rings of DNA or plasmids can be found in some bacteria cells. So just so you can notice here, we have some plasmid. This is the bacterium, and this will be the bacterium chromosomes. So number two, scientists remove the plasmid. An enzyme cuts open the plasmid and removes the human insulin gene from its chromosome. All right. Just like you can notice right over here, we are cutting the plasmid and we are removing the information of the insulin from the DNA of humans. Number three, the human insulin gene attaches to the open ends of the plasmid to form a closed ring. All right, so just like you can notice, at first we open the plasmid in the bacteria, and now we are inserting the part of the insulin to complete the ring. Number four, some bacterial cells take up the plasmids that have the insulin gene. All right, so one more time, we already have a complete circle inside the bacteria. So now what's going to happen? Number five, when the cells reproduce, the new cell will contain copies of the engineering plasmid. The foreign gene directs the cell to produce human insulin. So, just like you can notice, the bacteria that already has the engineering plasmid inside is going to start reproducing along with this different variation. So, since we inserted the insulin gene inside the bacteria, then way more insulin is going to be produced, which later can be used as medicine to fight diabetes. So, that'll be it for today's lesson, guys. It's been great to have you all around here, and I will see you during the mid session that will start in a couple of minutes. So, it's been great to see you all, and goodbye. I will see you next week.